Again, we're going to hit how organelles contribute to the function, but then we're really going to focus on how chloroplasts and mitochondria capture, store, and use energy. So, eukaryotes have membrane-bound organelles, including the nucleus and nucleolus. The nucleus stores genes with the chromatin. The nucleolus synthesizes ribosomes. The rough endoplasmic reticulum and the ribosomes are where proteins are synthesized or made, and then they are processed in the Golgi complex, modified and directed towards targets. Lysosomes contain hydrolytic enzymes which recycle old organelles and also break down organic compounds that are taken into the cell. The smooth ER is going to detoxify drugs and synthesize lipids. The mitochondria is important in converting the energy from glucose or other organic molecules into energy in the form of ATP. And then we have the centrosome and centrioles that are important in directing microtubules in the cytoskeleton and cell division. Ultimately, we end up with this compartmentalized cell, and it's important to understand that this is a generalized model, and cells in different parts of your body look very differently depending on the functions, and therefore, because they have different functions, their organelles are going to exist in different proportions. For example, a plasma cell within your immune system that's mass producing protein antibodies would have a lot more rough ER. Cells in your liver, which are important for detoxifying drugs and alcohol, would have more smooth ER. The liver would also have more smooth ER because it is important for making lipoproteins, which send out cholesterol. And that cholesterol is a precursor to steroid hormones. So your testes would also have a higher proportion of smooth ER because they are going to make the steroid hormone and synthesize testosterone. Muscle cells, which require a lot of ATP for muscle contraction, is going to have a lot more mitochondria. Now, plant cells, again, there's some differences here. Plant cells have a cell wall, which is important for supporting the overall shape of the cell. It protects it. It's going to prevent the lysing of the cell or the breaking of the cell through an increase in water. And that water is going to exist a lot in the central vacuole, but the central vacuole will help maintain that turgor pressure where the membrane pushes against the cell wall. It will also be a kind of a dumping ground for waste products because plant cells do not have lysosomes, but plant cells also have chloroplasts, and it's in that chloroplast that energy from the sun is captured and stored in carbon compounds. Plant cells also have the nucleus, which stores DNA and genes, and the nucleolus, which makes ribosomes. Also is going to have rough ER, again, synthesizing proteins, Golgi complex, which is modifying those proteins and directing them to their targets. The smooth ER is going to synthesize lipids. The mitochondria is going to process and organic compounds in order to take the energy from the bonds of those compounds and convert it into the energy in the bonds of ATP. And it does not have centrioles, but it still has a centrosome, which is important for the entire cytoskeleton and microtubules. Again, we have compartmentalization within eukaryotes. Now, chloroplasts and mitochondria are very important for capturing, storing, and using energy. So when we talk about energy, we're talking about mitochondria and chloroplasts. Mitochondria and chloroplasts. So within our chloroplasts, we have double membranes. We have thylakoids, stacks of thylakoids where the light reactions are going to take place. and all the space that's outside of the thylakoids is called the stroma, where the Calvin cycle occurs. So it's within these thylakoids where you have chlorophyll, which captures the energy from the sun. But water breaks down, electrons from the water go to the chlorophyll, energy from the sunlight energizes the electrons. Electrons power pumps where protons will get pumped inside the thylakoid space. Those electrons will be re-energized by the light and will get picked up by NAD to carry the high energy electrons with NADPH to the Calvin cycle. This concentration gradient created here by protons or hydrogen ions will ultimately have the protons diffuse out through ATP synthase. At that point, ATP will be created. The energy from ATP and NADPH will be put into carbon dioxide, and ultimately in the Calvin cycle, carbon dioxide is converted into G3P. And G3P is the precursor to fatty acids. It's the precursor to amino acids, and usually we think about photosynthesis creating glucose. If you put glucose and fructose together, you get sucrose. Glucose together can make starch or it can make cellulose. Now, eating plants and getting those organic molecules is ultimately going to be able to convert that energy in the bonds of the sugars back into energy in the bonds of ATP. But each of our macromolecules, all of our proteins, carbohydrates, and fats can all end up being used for energy depending on environmental needs. Only glucose goes through glycolysis, but we can take acetyl-CoA and get into the citric acid cycle within the mitochondrial matrix. And it is that citric acid cycle that is going to ultimately produce the carbon dioxide, but it's going to carry a lot of high energy electrons through NADH and FADH2. Those electron carriers are going to drop off these high energy electrons, which power the pumps, which is going to move hydrogen ions out of the matrix into the intermembrane space. And this concentration gradient is going to cause the protons to flow back in through ATP synthase, which is going to generate our ATP, which can be used for cellular work. At the very end here, oxygen is going to clean the electrons off of these pumps and pick up some hydrogen ions or protons and ultimately produce water. So we've got chloroplasts, we've got mitochondria. They're both going to create ATP, and they are both essential to capturing, storing, and using energy.